Good afternoon, everyone.
Good afternoon.
Okay. So, did you all go through the video which I had put on the Thursday's class?
No, sir, I'm facing the same problem.
What was that?
Sir, I'm not able to on that site.
Okay, is it so? I don't think it's like I have given you the link of that particular video. It should play, I suppose.
Sir, I don't
Anybody
know.
else?
I'm facing the problem.
You could not play?
No, sir.
So copy the link and place it in the different browser in your whatever mobile or whatever it is. Copy the link and then start
I
playing
have
it.
tried, sir.
Right, but you could. Anybody else facing the same problem? The ribosome one, which I had posted on Thursday.
I will try it once, sir, again and tell you about it.
Okay, do it then. Okay. I don't think it should be a problem because it's like I have given you the same link which is there on that particular video. I haven't made it as my own, so there shouldn't be any issue, I suppose. If at all, if it is not playing by clicking the link, and copy the link and go to your next browser, the fresh browser, and there you open it. Try opening it. Anybody
Okay,
else
sir,
is
I'll
facing
try.
this? Okay. Anybody else is facing the same type of problem? No. किसी और को प्रॉब्लम हो रही थी ऐसे? बाकी सब का कोई आए हैं कि नहीं आए हैं? हमें आते हैं. Only 10 people get into the class. Okay, today we will discuss about something different, which is not a type of any organelle. It's a structure which is being formed when the cell divides. You know, cell divides in two ways. You must have gone through that cell cycle, which tells you about the different phases a cell goes in his life tenure. So first, suppose if it has divided, it will go to the resting phase and it will grow. It will it will develop and grow, and then it will reach to a stage where again it is able to divide itself. And for that, before getting into the cell division process, it usually prepares itself. It grows to a bigger size, matured size, and then it replicates its genetic material. That is we call as phase or synthesis phase. From there again it grows a little bit and it stays for some time, and then it enters into M phase. That is cell division state, either mitotic division or the meiosis. So you all know meiotic and mitotic. Mitotic means it's an equational division where the number of chromosomes or the genetic material is same in the daughter cells, but in directional division or the meiotic division, the genetic material is being reduced to half, and then it mitotically divides to further accomplish its function. So meiosis or the meiotic division usually happens in the germ cells where they are required to make gametes, right? Because the gametes unite to form a zygote, and from the life begins. From there the life begins. So the gametes need to have half the number of the chromosomes, or half the quantity of the genetic material which has to be transferred to the next generation. So that way they are usually done with they are seen in the germ cells where the gametes are formed, because gametes unite. That's why they carry the half of the chromosomes. That means we all know exactly. Usually we know that we are diploid type of animals. That means we have got two sets of genetic material, or you can say two sets of chromosomes, which carry the DNA molecule and the information and the genes and all the traits and characters responsible for the expression of them. So the code for that, the codes are carried by these chromosomes, which are usually in two sets in every individual. That's why we call them as diploid, right? Diploidy is the set of chromosomes, and the diploidy means there are two sets of them. And when they have been reduced to half, that means one set of the chromosomes are there in each gamete. So there we call them as haploid. That means half of the diploid sets of chromosomes. So the haploid set, you would say the haploid set is nothing but the one second. Haploid set is nothing but the one set which constitutes all the genes and all the traits of the characters and the codes for them, which are responsible to be expressed. But for evolution point of view, or for the improvisation point of view, another set comes from another gamete of different sex, and then they unite to form a zygote where they have got two sets of the same set of chromosomes, or same set of genes or alleles or traits, codes for the traits, which are to be transferred to the next generation. And these two sets would improve in the expression of the particular gene. And that expression of the particular gene or the allele or the code for the trait are usually improvised, or they usually follow the principles of our father of genetics called Mendel, right? Gregor Mendel. So the Mendelian principles are followed. The postulates established by Gregor Mendel are followed to get themselves improvised or more evolved or more developed sort of expression of the genes or the codes of traits and characters. So in mitosis, it's very simple that at the time of division, when the cell reaches to this time of division, the whole genetic material gets condensed and forms thread-like structures called chromosomes. And those chromosomes are usually with the help of astral fibers, which are formed at the time of division. And now the phases of the division, these chromosomes are, they have been placed on the equatorial plate, the center of uh, the cell. But how does the equatorial plane is confirmed? Because the centrosome, which contains a pair of centrioles, the centrioles, they move apart to the opposite ends, and then those opposite ends would, demarcate the what is the equatorial line, and the chromosomes get arranged themselves in the equatorial line. That phase is called, you all must be knowing what are those phases. So they settle down and that they place themselves at the equatorial plane, and from there the centriole extends its the fibrous structures which come and hold the chromosomes, and they try to separate them. We all usually see the chromosome X, that means it's been duplicated. The chromosome has been duplicated, and these duplicated chromosomes are now separated with the help of astral fibers, which have been, get attached and extended from the centriole of the centrosome, and those centrioles which have been, moved apart onto the opposite ends for this purpose. So that way we usually know what exactly how the cell division in a normal sense, or the mitotic division, is carried on. And now comes the, there's a typical another type of cell division that's called meiosis. Meiosis is also called the reductional cell division, in which the content is been reduced to half. Why this is being reduced to half? Because they are used in the creation of gametes, which usually unite and form a zygote. From there, they get into the development of a new individual, right? So these gametes, which have got half the number of the chromosomes in them, they are usually that stage of that particular cells are made with the help of a division called meiosis, and this is called reductional division 2. So what happens in this? In the first phase, where we usually call the cell division, we usually categorize the cell division in different phases: prophase, metaphase, anaphase, and telophase. So that way, what happens in prophase, you need to study if at all you have forgotten, because you must have already read about it. So they get settled, the kind of chromosomes are settled, and they are condensed, and they form the nuclear material is condensed, and they form chromosomes. That's in the prophase. And then from there they go to the placement of all those chromosomes at the equatorial plane, and the centrioles start moving to the opposite side. That's called the metaphase. And then the astral fibers extend from the centrioles, and they get attached to the equatorially placed chromosomes, and that is called the anaphase. And the telophase is the portion in which the chromosomes, those duplicated chromosomes, are separated out, and you could find the two sets of chromosomes at two different places near the centrioles. So that's a telophase. And from there the dikinesis happens. The cytoplasm gets invaginated, and the two separate cells, smaller cells, are formed, and in the meantime, simultaneously, the chromosomes, they get, they enter into the resting phase, and the chromosomes get decondensed, and the nucleic material is again seen, which a membrane called nuclear membrane. So that way the cell gets divided into it's a normal kind of division, normal way of division, in which the chromosome number or the genetic material is being conserved. Whatever chromosome, whatever genetic material, nucleoplasm, or the chromatin is there in a cell, that's been conserved, because it's been imparted, it's been transferred to, it's been inherited to the new cells. So that conservation is nothing but the cell being following the synthesis phase and getting into the division phase. The chromosomes, the genetic material is being duplicated, and the duplicate set is being, transferred to the next cell. But in meiosis, what happens in the first phase, that is the prophase, when the chromosomes are seen, when the genetic material or the nucleic material is been condensed and forming the chromosomes specific to any individual, any organism. So those chromosomes are now have some sub phase stages. That means in the prophase only, it undergoes a type of multiple phasic divisions in which it forms some few structures, new structures, and they get half of the number of the chromosomes. Now suppose, as we have seen in mitotic division, the chromatic material or the nucleic material or the genetic material which has to be transferred, it condenses into the chromosomes. As we have said, the animal is diploid, that means diploid, that means two sets of chromosomes are there, one which have been received from the male gamete and the another which has been received from the female gamete. That way, the half of the chromosomes have been received from the mother and half from the father. That way we have got two sets of chromosomes. So these two sets of chromosomes get duplicated. That means the four sets are formed and two separate sets are been bifurcated or the separated out into two different cells in mitotic division. But in meiosis, what happens? In meiosis, what happens? The homologous pairs come together at the sub-phasic divisions, sub-phasic levels. That means the prophase has got different phases to reveal or to display. And in that, what happens? The chromosomes, they have been multiplied. They have been synthesized before entering into cell division. The material chromosomes or the genetic material have to duplicate it. They've been duplicated, but they are not separated out by the, as in anaphase it happens, the astral fibers come and separate them out. But in the initial stage only, in the prophase phase only, when the chromosomes are condensed, they usually get themselves, paired up. Like the chromosome which has been transferred from the male, it will pair from the same type of the chromosome which has been transferred from the female. So they are called homologous chromosomes. So these pairs, they form pairs, they come together, and that particular coming to one stage, there are different names for these phases also. When they come together, when they form a pair of homologous chromosomes together, they stay for some time, and that particular sub phase is called packeting stage. In that, those homologous chromosomes stay together, and there are two types of phenomena they occur, that is called crossing over or the linkage. So the linkage and crossing over. Now we need to understand where, when the chromosomes are when the chromatin is being condensed to form chromosomes, the homologous pairs get together, they come near, and they form a pair. And that pair would, it would not stay for a longer time if something is not binding them, right? So something has to be bound to make them stay for a longer time, so that they can have the phenomena of crossing over. Crossing over means the exchange of the genetic material from one chromosome to another. That means the genetic material which is there in the chromosome 1 which has been derived from the mother will have some exchange of its matter from the chromosome 1 of the one which has been received from the father. That way, the crossing over, the physical exchange of the chromatin material from chromosome 1 to another, which are homologous, is something called crossing over. And there's another thing which is called linkage. Linkage is the effect of that particular portion of the chromosome is affected, or it's been, it's influenced by the same type of the same chromosome of the homologous nature will have the same chromosome, but it will have the influence of that particular chromosome on itself, and it will express itself in a different way. The expression might change. That's because of the same type of chromosome present on the homologous pair. So that way, in meiosis, we have got, in the prophase only, we have got multiple stages in which they get condensed and the homologous pairs come together. And now coming together is okay, and if the duplication has already been done in the S phase of the cell cycle, they are now duplicated, but they are not separated. Those X type of visible chromosome of coming from mother will pair with the coming X type of chromosome of the father, but they have to be bound together. They have to be held so that they would stay near and in the pair, so that the physical transfer of genetic material might be carried on. So for the to carry on that particular process, they are to be there in the near vicinity, and keeping them in near vicinity, keeping them in pair, we need something to bind them, and that something is nothing but synaptonemal complex, synaptonemal attachment, synaptonemal attachments. So what happens when a chromosome is, when it is duplicated, it looks like X. So the chromatins, which are like looking like this, these chromatins are called sister chromatins because they are of the same chromosome. If it is the chromatin of the different chromosomes, suppose there is one chromosome which has got two chromatins, and is another chromosome which has got two chromatins. You know, this one, the moving hand. These are the sister chromatids. On these fingers, two fingers, sister chromatids, and here they are the sister chromatids of this particular chromosome. But as this arm, this middle finger, and this middle finger of the two different chromosomes are called non-sister chromatids. These are non-sister chromatids, these are non-sister chromatids, but these two are sister chromatids of respective chromosomes. So that way, the non-sister chromatids would take the, physically they might exchange the chromatin or the portion of some chromosome, so that the future generation might get some improvements, or might get some advancement in the expression of that particular chromatin or the chromosome or the gene present on that particular portion of the chromosome. So for the improvisation, for the development, for the betterment of the expression of that particular gene, these crossing overs take place. So for the crossing over purpose, they need to be placed together for some time. They need to be placed together for some time, and that structure which holds them for some time to get into a process called crossing over or the linkage, they need to stay together for some time, and that's been done by a structure which is called synaptonemal complex. And the synaptonemal comes up, this is the chromosome, this is another chromosome, and these two chromosomes are held together with the synaptonemal complex, which is nothing but a structure of proteinaceous nature, which is the structure of proteins, and which could bind these two chromatids of non-sister chromatids. When there is a binding or an attachment with some proteins within the non-sister chromatins of the two homologous pairs, that is called the synaptonemal complex. They have been, it's been highly, very extensively studied, and people know what type of proteins are there. So usually these two chromatins are, when they are bound together, there is a, interlinking protein molecules. They are called the zip-like, zip in zip में जैसे होते हैं ना ऐसे lines दिखते हैं, so there are zip-like proteins which help them holding these non-sister chromatids of two homologous pairs of chromosomes. So that way, तो zip-like जो structure के proteins बनते हैं, और उसके different types of they have zip proteins कहा जाता है उन्हें. इसके अलावा भी there are synaptonemal complex proteins हैं, SCPs हैं, उसके भी types हैं, and वो कैसे form होते हैं, how they are formed, and क्योंकि zip में भी zip 1, zip 2, zip 3, three types of zip proteins are there. Zip 1 helps in cross-linking these chromatids, and I think zip 3 or zip 2 helps in dissolving these cross-links. The cross-linking would help these chromatins stay together for any kind of crossing over, right? तो क्रॉसिंग ओवर होने के लिए उनको कुछ समय तक साथ रहना होता है, एंड वही साथ रहने के लिए ये कॉम्प्लेक्सेस फॉर्म होते हैं, जो कि यूजुअली प्रोटीन्स हैं, तो हम इनको न्यूक्लियोप्रोटीन स्ट्रक्चर्स भी कहते हैं. यही सिनाप्टोनिमल एक प्रकार का सिनाप्स है, जोड़ है. ये सिनाप्स मतलब अटैचमेंट, जोड़, जंक्शन. तो एक प्रकार का जंक्शन है where the crossing over might take place. So that way, the main purpose of synaptonemal complex is to make these homologous pairs. अभी chromosomes duplicate हो चुके हैं, यानी सपोज करिए हम ह्यूमन के केस में कंसिडर कर रहे हैं, जैसे माइटोसिस था, तो माइटोसिस में कैसे 46 क्रोमोसोम्स हैं, तो 46 के वो फिर कितने हो जाएंगे, 46 क्रोमोसोम्स ही है ना अपने पास, तो कितने हो जाएंगे वो 92 क्रोमोसोम्स बन जाएंगे। लेकिन वो जो 46 है, जो एक का जो दो हुआ है, वो दोनों अलग नहीं होते, उनको अलग करता है जो एस्ट्रल फाइबर्स जो आते हैं सेंट्रियोल से, वो खींच के दोनों को अलग कर देते हैं। तो 92 में से फिर एक तरफ 46 हो गए, एक तरफ 46 हो गए, डुप्लीकेशन ऑफ मटेरियल। अब यहाँ पे क्या हो रहा है, यहाँ पे डुप्लीकेट जरूर हो चुके हैं, दे हैव बिकम 92, बट लेकिन वो सेपरेट नहीं हुए हैं। और उसमें से भी क्या हो रहा है कि वो होमोलोगस पेयर जो है साथ में आते हैं। अब नाउ एस्ट्रल फाइबर्स जो है, जैसे कि माइटोटिक डिवीजन में वो खींच के अलग किए थे, वैसे ही ये जब ये होमोलोगस क्रोमोसोम साथ में आ चुके थे जो डुप्लीकेटेड है, उनको फिर वो अलग कर देते हैं। यानी कि एक सेट एक तरफ चला जाता है, एक सेट एक तरफ चला जाता है। यानी कि जो 46 क्रोमोसोम है उसमें से 23 का सेट एक था मेल वाला फादर की तरफ से, 23 सेट मदर की तरफ से था। ये दोनों साथ में आ जाते हैं जस्ट फॉर द प्रिपेयरिंग द सिनाप्टोनिमल कॉम्प्लेक्स, और सिनाप्टोनिमल कॉम्प्लेक्स उनको हेल्प करता है फॉर द एक्सचेंज ऑफ मटेरियल। लेकिन ये 4 हो गए साथ में, 2 इधर होना चाहिए, तो वैसे ये 2 इधर हो जाते हैं। लेकिन क्या हो गया, इसके जो 2 हुए तो वो दोनों अलग नहीं हुए, वो साथ में ही है। और जो होमोलोगस पेयर में जो दूसरा पेयर था, पेयर अलग हो गया, यानी कि पेयर टूट जाता है। 46 क्रोमोसोम्स हाफ होके 23 एक तरफ चले जाते हैं, 23 एक तरफ चले जाते हैं, राइट। फिर उसके बाद वो सेपरेट हो जाएंगे एंड वो फिर आगे गमेट्स के फॉर्मेशन में आ जाते हैं। अब यहाँ से उसके बाद वो माइटोटिक डिवीजन नहीं होता उनका, सेल प्रोलिफरेशन होता है। सेल प्रोलिफरेशन को हम कहते हैं यूजुअली, जैसे कि आई मीन एक डिवीजन हो गया, उसके बाद फिर बाद में वो जैसे एग वाली केस में देखा जाए तो एक के 4 बनते हैं, तो उसमें से एक ही एक रिडक्शन डिवीजन होता है। कैसे कि वो 46 क्रोमोसोम से 23 अलग हो गए, वो फिर मियोटिक के फर्स्ट फेज के डिवीजन के पोर्शन है। उसके बाद फिर वो दोनों अलग अलग होकर चले जाते हैं, 2 के 4 बन जाते हैं। तो 2 के 4 बने, लेकिन उसमें कितना हुआ 23 डुप्लीकेट हो चुका था, 23 डुप्लीकेट हो चुके थे, 23 डुप्लीकेट हो चुके थे। लेकिन ये सब के सब 2 इसके अलग नहीं होते, ये होमोलोगस पेयर्स अलग हो जाते हैं। एंड देन फिर इनके फिर डुप्लीकेटेड वाले पोर्शन अलग होते हैं। तो कैसा हुआ, हमारे पास 23 क्रोमोसोम्स ही है रिजल्टेंट, ये 4 सेल्स बन जाते हैं रिडक्शन डिवीजन के बाद। एंड दैट टू एग फॉर्मेशन में यूजुअली ऐसा होता है कि उनमें से 4 में से सिर्फ एक ही है जो एग जो है फॉलिकल बनता है, जो मैच्योर होता है, बाकी तीनों पोलर बॉडीज जैसे काम करते हैं। अब वैसे ही मतलब उनका फर्दर डिवीजन नहीं होता, माइटोटिक डिवीजन होता है। लेकिन इन केस ऑफ स्पर्म में भी सेम वैसा ही होगा, लेकिन चारों जो सेल्स हैं वो चारों सेल्स प्रोलिफरेशन के अंदर प्रोसेस में एंटर करते हैं, इसको हम बोलते हैं क्लोनल सिलेक्शन। वो अपने डुप्लीकेट्स बनाते हैं, दैट्स क्लोनिंग कहते हैं, एक प्रकार का क्लोनिंग होता है, और ढेर सारे बन जाता है। एक्स वाले स्पर्म होंगे तो ढेर सारे एक्स वाले स्पर्म्स बन जाएंगे, वाई वाले होंगे तो ढेर सारे वाई वाले स्पर्म बन जाएंगे, बिकॉज दे नीड टू बी मोर इन क्वांटिटी, बिकॉज दे हैव अ टारगेट ऑफ 1 एग। सो वो एक अलग फिलॉसफी है। तो दैट वे कि रिडक्शनल डिविजन्स में जब होमोलोगस पेयर्स एक प्रोसेस को सपोर्ट करते हैं, वो उस प्रोसेस को सपोर्ट करना है मतलब क्रॉसिंग ओवर को, तो क्रॉसिंग ओवर होने के लिए होमोलोगस पेयर्स ऑफ क्रोमोसोम्स साथ में होने चाहिए फॉर सम टाइम। इसलिए पैकेटिंग स्टेज जो है थोड़ी सी लॉन्गेस्ट स्टेज है, जिसमें वो होमोलोगस क्रोमोसोम्स साथ में रहते हैं, और उसे हम कहते हैं, एक और नाम है उसका जिसका नाम है टेट्राड स्टेज मतलब कि जो क्रोमोसोम था, एक क्रोमोसोम उसका होमोलोगस पेयर दूसरा क्रोमोसोम, ये दोनों के इसके भी 2 क्रोमैटिड्स हो गए, इसके भी 2 क्रोमैटिड्स हो गए, तो ये दोनों 2 और 2 4 दिखते हैं एक साथ, एंड दे आर हेल्ड विद द सिनाप्टोनिमल कॉम्प्लेक्स, जिप लाइक जो प्रोटीन्स है वो उनको होल्ड करते हैं। तो क्या होता है कि एक समय में आप देखोगे एक मोटा सा कुछ तो भी क्रोमोसोम नजर आ रहा है व्हिच हैज गॉट 4 क्रोमैटिड्स, इसलिए इसको टेट्राड स्टेज बोला गया था। और हर क्रोमोसोम जब सिनाप्टोनिमल कॉम्प्लेक्स फॉर्म करता है, तो ये जो एक्स है इसके ऊपर जिप प्रोटीन्स लगे होते हैं, तो उसमें से ऐसे क्रॉस लिंक्ड वाले ना प्रोटीन्स लगे हुए थे, उसको काइज़मा बोला जाता है, एक्स फॉर्मेशन। और उसमें जो है जैसे मेरी हाथ की लाइंस दिख रही है, तो ऐसे ये पूरे एक्स पे क्रोमोसोम पे वो क्रॉस लिंक करने के लिए, इससे सिनाप्टोनिमल कॉम्प्लेक्स फॉर्म करने के लिए जो जिप प्रोटीन्स लगते हैं इसके ऊपर अटैच होते हैं, वो ऐसा लगता है कि एक ऐसे एक्स बना हुआ है, उस पे ऐसे क्रॉस लिंक लाइंस बनी हुई है। तो वो भी ऑब्जर्व किया था, इसलिए उसको कायजमा बोला गया था, कायजमाटा भी कहते हैं उसको। तो एक कायजमाटा एक क्रोमोसोम का, एक होमोलोगस का, और कायजमाटा दूसरा क्रोमोसोम, ये दोनों साथ में जमाएंगे तो कायजमा जो है जोड़ लेते हैं और वो फॉर्म करता है सिनेप्टोनिमल कॉम्प्लेक्स। तो एक सिग्निफिकेंट सिनेप्टोनिमल कॉम्प्लेक्स का ये है, मैंने एक वीडियो अपलोड की है अभी, आप लोग यू कैन गो थ्रू इट, यू वुड अंडरस्टैंड हाउ एग्जैक्टली। हालांकि उसमें प्रोटीन्स के बारे में भी ज़्यादा बताया कि व्हाट आर द डिफरेंट टाइप्स ऑफ प्रोटीन्स which help in forming those zip-like cross-linking structures, और कौन से प्रोटीन्स हैं जो when the thing is over, like the packaging का टाइम हो गया, क्रॉसिंग ओवर हो चुका है, then they get separated, तो zips को डिसॉल्व करना होता है, काइज़मा को डिसॉल्व करना होता है। तो डिसॉल्व करने के लिए भी कुछ प्रोटीन मॉलिक्यूल्स हैं, उसके बारे में भी उसमें जिक्र किया है। आई थिंक मेरी आवाज़ आ रही थी अभी तक कि मैं ऐसे बड़बड़ करे जा रहा हूँ, सुनाई पड़ रहा था आप लोगों को?
Yes, sir, आ रही है.
Okay. So that's all for today. We would not go much like into details because आप लोग को प्रोटीन्स के बारे में उतना ज़्यादा जानकारी रखने की ज़रूरत नहीं है. But still, I have posted a video which is an extensive elaborative way of explaining the synaptonimal complex, जिसमें कौन से प्रोटीन मॉलिक्यूल्स हैं, कौन से जिप प्रोटीन्स हैं जो हेल्प करते हैं जिप 1, जिप 2, जिप 3 जो हैं, और कौन से जो प्रोटीन्स हैं जो उनको फॉर्म करने में मदद करते हैं, और कौन से प्रोटीन्स में एक सुमो नाम का भी प्रोटीन आता है, वो उसका यूबिक्विटोन का स्मॉल यूबिक्विटोन मॉडिफायर, एक मॉडिफायर है जो मतलब जिप प्रोटीन्स को, जैसे मैंने बताया ना, एक काइज़मा फॉर्म हो गया, यानी कि एक डुप्लीकेट हो चुका है, होमोलोगस पेयर का एक है, इसमें जैसे मैंने लाइंस बताई वैसे जिप प्रोटीन से अटैच होते हैं। जब जिप प्रोटीन से अटैच होते हैं तो जिप प्रोटीन से दूसरे वाले से कैसे अटैच होंगे फिर? तो छोटे दूसरे प्रोटीन मॉलिक्यूल्स होते हैं जिनको सुमो मॉलिक्यूल्स कहते हैं। तो वो सुमो मॉलिक्यूल्स दोनों को जोड़ देते हैं जब सिनेप्टोनिमल कॉम्प्लेक्स एस्टैब्लिश हो जाता है। तो सुमो मॉलिक्यूल्स को फिर से ब्रेक करने के लिए फिर जिप 3 है, शायद जिप 3 मॉलिक्यूल है जो उनको फिर डिसॉल्व कर देता है। दिस इज ऑल to make them close enough for a longer period so that crossing over might take place. The basic purpose उनका वही है। So that's all for today. वीडियो को देखिए एक बार and try understanding things. That's all. Okay. Thank you for today. Thank you. We'll sign off.
Thank you, sir.
Okay.